Welcome. The neural response to food pictures involves a region of the dorsal mid-insula, a region which also exhibits a reliable response to actual taste, suggesting the possibility that these responses might reflect the inferred taste of these depicted foods. In one recent study, we used ultra-high resolution fMRI and multivoxel pattern analysis, and we were able to classify the neural response to liquid taste scents delivered during scanning, as well as a response to food pictures grouped by their dominant tastes within this region. However, our cross-task classification analysis was unsuccessful, as we found that the patterns for food pictures and tastes were actually unrelated. There are multiple possibilities for this result, one of them being that these food picture responses might simply represent different properties than the taste of these foods, such as shape, color, or texture. Another possibility could be that differences between the tasks that were experiment-related, such as swallowing-induced motion during the taste task, might have led to these differences. Or it may simply be that food pictures and actual tastes activate different populations of neurons within the mid-insula. To address these issues, we performed a follow-up study where we had 22 subjects undergo 7T fMRI while performing one task where they imagined specific basic tastes during scanning, and another where they viewed pictures of foods and non-food objects. For the taste imagining task, we prompted them with the words sugar, salt, lemon juice, and water during scanning, indicating that they were to imagine the taste of these substances placed on their tongue. In the second task, they saw pictures of specific sweet, sour, or salty foods, and they also saw pictures of several types of non-food items serving as a control stimulus. During half the picture blocks, one of the pictures in the block was repeated, and they were told to press a button to indicate when they saw the repeat. For our univariate analysis of the taste imagining task, we contrasted the imagined taste of sugar, salt, and lemon juice versus the imagined taste of water, and we identified strong bilateral activation of the dorsal mid-insula and oral somatosensory cortex, brain regions that are consistently activated during actual taste experience. We next ran multivariate searchlight analyses on the data from both tasks and performed a union of the decoding maps from these tasks, which highlighted the left mid-insula and the ventral visual cortex as regions where imagined tastes and food picture categories could be accurately decoded. Finally, we performed a cross-task classification searchlight, where we trained on data from the imagining task and tested on data from the food picture task and vice versa. And this analysis specifically identified the dorsal mid-insula as an area where we could reliably cross-classify imagined tastes and food pictures. For our final analysis, we ran a pattern similarity analysis within a taste-responsive region of interest from previous study, comparing the average spatial patterns of our imagined tastes and food picture categories. As expected, the patterns across these two tasks were reliably similar, as well as taste quality specific, as the similarity for pairings of the same imagined taste and food picture type were significantly greater than the pairings of different tastes and food pictures. So overall, these results suggest that imagining a specific taste activates the brain regions involved in taste experience, and as the food pictures and imagined basic tastes evoked reliably similar patterns of activation within the mid-insula, our results also seem to signify that yes, these food picture responses do indeed represent the inferred taste of these foods. And in keeping with previous studies of visual cortex, these results also suggest that this region supports a common neural coding scheme shared across voluntary and automatic gustatory mental imagery. In conclusion, I'd like to thank my PI Alex Martin and my trainee Maddie Carrington, who helped inspire and carry out this research project. Please scan the QR code if you're interested in a preprint of our manuscript.